Now we will be heading towards the next session. In our next session, now I call upon Dr. Ingole sir on the dais to chair the next session. Dr. Rajesh Karyakarte and Dr. Ingole sir to chair the next session. Dr. Ingole is the professor and head in the Department of Microbiology, GMC, Jalgaon. I request Dr. Rajesh Karyakarte to introduce Dr. Anurag Agarwal.
Request that you will have lunch after this lecture. That is a request to all of you. It's an honor for me to introduce Professor Anurag Agarwal. He is the Dean, Bioscience and Health Research, Trivedi School of Bioscience, Asoka University, India, and former Director of Institute of Genomics and Integrative Biology, a National Laboratory of CSIR, India. After completing graduate medical education at All India Institute of Medical Sciences, Delhi, he further trained in internal medicine, pulmonary medicine, and critical care at Baylor College of Medicine, Houston, USA, followed by a PhD in physiology from Delhi University. His primary research is in respiratory biology, and broader interests are in new vision of health and healthcare seen through the lens of emerging technologies. He serves on numerous national and global advisory groups, recently chairing the World Health Organization. Please remember, please try to understand this very important thing. He chairs the World Health Organization Technical Advisory Group for SARS-CoV-2 viral evolution. So he names the variants. And Lancet Financial Times Commission for Governing Digital Health Futures. I think this is the key which you like, the digital health futures, and serving on the pandemic preparedness subgroup at Global Partnership for Artificial Intelligence. He received the Shanti Swarup Bhatnagar Prize in 2014 and Sun Pharma Foundation Award in 2020. He is the Fellow of Indian National Science Academies. I welcome you, sir, for your talk.
So I have the job of Hello? Yeah, great. So I'm standing between you and lunch. So I'll try to make this as fast and informative as I can. You heard a lot today about variants. What I'm going to take it to is a natural conclusion, the vaccines, the way to end the problem. This picture is from a previous century. The Japanese schoolgirls wearing a mask while going to school, people raiding saloons for the war on influenza, people telling people to keep church windows open. The more things change, the more they remain the same. 100 years later, we are still there. So, influenza, despite having been with us for thousands of years, is able to create pandemics. So can other viruses. With SARS-CoV-2, what we saw, a respiratory virus with minimal prior immunity got into human circulation. We can speculate on how, as long as we want, it won't matter. It evolved very fast to even fitter variants. The D614G mutation came very early in the pandemic. That's pretty much the index now. Spread all over the world. Airborne transmissibility was not recognized until a bit later than it should have been. And our hopes that because of immunity, we will get herd immunity, it will stop, never materialized because the virus kept evolving, kept evading immunity, kept creating fresh outbreaks, and with every outbreak, although it appeared the disease became milder sometimes, the number of infections kept giving the virus an opportunity to evolve further. So these massive surges overwhelmed healthcare in the beginning. Later on was a little bit different. So let me walk us through that. Could we have known about the Delta wave and what happened by looking at the experience of the rest of the world? Perhaps we could. If you take the case of Manaus, Brazil, you will notice that by October, November in 2020, they had reached a seropositivity of approximately 60% in blood samples taken from blood banks. They had opened up the country, they had not suffered any consequences, life was back to normal, and suddenly, in December 2020 to January 2021, cases started rising again. They ran out of oxygens, wards were full, innovative things of splitting a cylinder into multiple outlets to take care of patients, those pictures were being circulated around the world. This is December 2020, January 2021. And P.1, gamma, would be the WHO word for it, was found to be responsible for this outbreak. By that time, alpha and beta had already been described in South Africa and UK. Gamma came creating new outbreaks in a highly immune country. There was actually no reason to believe that anybody was safe. Okay, let me figure this out. In this context, in February 2021, there was a feeling in India that we were done. These were numbers. It's a tweet from Delhi talking about the 5th sero survey done in the city with 56.13% of the population showing antibodies, and this is before the vaccination really started. Cases were down after the initial waves, and we also thought that perhaps we were done. That thinking, that learning from Manaus would have been good for us. And of course, you know how the pandemic went, but before that, let me walk you through what Dr. Karyakarte and his team found in February 2021. In February 2021, the BGMC team, middle of the month, described for the first time a variant with new mutations. Over the next 2 to 3 weeks, we had this picture. We could see that particular variant was rising. We could see the cases were rising. And there was a discussion at that point whether there was a problem with Maharashtra's ability to control the outbreak, or was there genuinely a new variant driving the outbreak. Looking back, one can look at this graph and say the answer was staring us in the face. But in any case, while BGMC, the work led by them, led to the description of Kappa as well as Delta, I will say this much. Most of us were surprised when Kappa, which contained the E484Q, versus Delta, which lost the E484Q, which one of them spread faster. Things became much more crystal clear in early April in Delhi. Now the virus has already left. So I'm showing you a group of doctors. Most of them are double vaccinated. The dark blue color is double vaccinated. The light blue color is non-vaccinated. The intermediate blue color is single vaccinated. All these doctors were from a single hospital in Delhi, Sir Gangaram Hospital. Given the advisories floating around the world saying that you don't need to wear masks if you have been vaccinated, they were having coffee together. Out of about 35 doctors, of whom 30 were vaccinated, 25 double vaccinated, 20 broke through. What you can see here in light green, it won't work very well on the screen. What you see here on light green is what is Delta. What you see in dark green is Kappa, and dark brown is ancestor strains. By this time, it became very obvious. If you have 30 double vaccinated doctors and 20 of them break through because of a new variant that is circulating, that variant is not to be taken lightly. But we have gone from February, the original description from BJMC, to April, to an outbreak in Delhi that is perhaps being taken a little bit more seriously, and we have lost 2 precious months. And of course, by the time we saw this, the pandemic had really started again, and this graph hopefully illustrates very clearly for you what happened next. What I want to show you in the bottom graph over here is hospitalizations rose. ICU admissions failed to rise at a particular point in time. Do you think it is because these were not needed? Of course not. It is simply because Delhi ran out of ICU beds. That plateau you see marked by the arrow is because there were no ICU beds left in the city of Delhi. Hospitalizations continued to rise, ICU admissions did not. And any of us who were in Delhi at that time, or in many other cities the month after, will recognize that particular time of the pandemic in Indian history. To my mind, the reason I bring it up, it's a cost that we paid for not having a well-developed surveillance system that everybody trusted and followed at the same time together. And this particular graph only shows what happened in Delhi, then spread all over the country, and pretty much state after state after state, that's what you saw. That light purple thing that you see is delta. The yellow actually is alpha. In the background immunity that Delhi had reached, alpha was barely able to prosper despite creating havoc in places like North America, in places like UK, because alpha was less likely to escape immune response to the ancestral strain. And what we learned very soon was that delta was different. Delta was different in the sense, and now you're looking at cultures from organoids on the right hand side, you're looking at CT values on the left hand side. What you see is the viral load seen by delta was unprecedented compared to previous variants. Another thing what we were able to see, which I think was very important, and I'm going to emphasize this point a little bit, is that we were never able to fully track the real course of the pandemic because we were always relying on cases. What I show you on the top, and it's a little hard on this, yeah, perhaps you'll see, it doesn't matter. What I show you on the top left graph is seropositivity in our cohort in Delhi, in whom we test the antibodies for blood in all people of our institute regularly. What I want you to see is on the extreme left, the seropositivity used to vary between rich people and poor people. You will notice I have people using private transport, you will notice I have people using public transport, you will notice I have faculty, you will notice I have contract workers, and you can see that rich people can protect themselves very well. And already by the end of 2020, we had reached 10-20% seropositivity, middle 2020. In the beginning of 2021, we had reached about 40% seropositivity. But the rich people were still down to 20-30%. 70-80% of rich people had not been infected at that point in time. Look at what the seropositivity was in my institute at the end of the Delta wave in Delhi, 90%. It no longer respected economic differences, whether you were rich, whether you were poor. 90% of the people living in Delhi had been infected. This is a massive number. And particularly this previously infected, older, middle class to richer group of people was what contributed to the deaths and destruction. The reason the wave ended was not because we did a great job suddenly in controlling it. The reason it ended in Delhi is because 90% of the people had been infected. Not just that, on the bottom left, I want you to see titers of antibodies. Now, I think on the screen it doesn't work. That's okay. I'll walk you through the slides by speaking. What you'll see in many people, the antibody levels were coming down. So look at the red dot compared to the initial greenish blackish dot. Then suddenly, the T3, it goes up again. These are reinfections. 20 to 30% of people in our cohort got reinfected. That's a large number. These are not the people who became very sick. These are people who were able to get reinfected and thereby break that little herd immunity chances that Delhi may have had at that point in time. What I want to emphasize now by switching gears is vaccination saves lives and data is not reliable. Two things at the same time. On the top axis in blue, you see cases. In the bottom, you see in red, you see deaths. They're on different scales, okay? The point is not that scale is different. The point is they are a reflection of each other. The country on top left is the United Kingdom. That vertical line is when UK manages to get its people vaccinated. Look at what happens after that. The blue goes up, the red is no longer the same reflection, it's flattened. You see the same thing in Portugal. Look at Namibia. Namibia does not get vaccines. Cases go up, deaths go up as they did before. Look at Tunisia. Look at South Africa. Look at Indonesia. All the countries that did not get vaccinated, the Delta wave comes and people die. They die in almost the same ratio as they died in the past because we do not measure the numerator correctly. It is impossible that the number of cases in Indonesia were this small on the same scale compared to the number of cases in UK. It is simply that they tested less, they found less cases, they found less deaths. Numbers are not reliable, patterns are. Whenever you want to consider the Indian data that we have, remember this. If we look at the official number of cases, we have 30,000 cases per million. Convert that to percentage, that is 3%. Sometimes we speak of our 30,000 per cases per million as evidence of better control of the pandemic in India. I just showed you a Delhi slide of ending at 90% seropositivity before vaccination. We are pretty much at 90% across this country. And to prove my point, and to illustrate something very important for Maharashtra, let me show you the results of our sero surveys across multiple CSR labs in the country. As you can see, light yellow is below 10%. As you can see, dark purplish is approaching 100%. These are three different time frames. The first is June to September 2020. Next is January to March 2021. And the last is May to August 2021. What you will see is, contrary to what appears by looking at number of cases in Maharashtra, when you look at seropositivity in CSIR labs of comparably sized cities across the country, Maharashtra is just following the national trend. In fact, in January-March 2021, Maharashtra and Kerala, which have the largest number of cases, are actually behind the national trend. And by the end of the Delta wave, they are far behind the national trend. So there was no state that did much better or much worse in terms of controlling the spread of the pandemic. Each got its waves at different points in time. Some measured it better than others, and they are the ones where it appears the pandemic was more severe. In fact, even if you were to look at deaths per million, what we are fighting about with WHO, if you were to simply take Kerala's data, Kerala has, I think, 70,000 deaths with about its population is 2,000 deaths per million. WHO's estimate for India is around 2,500 deaths per million. 2,000 per million and 2,500 per million is not very different. And we know that Kerala never had any oxygen shortages. It is, of course, a higher longevity state. There are more old people. But when you look at Kerala at 2,000 per million without any major incidences, you look at a national number of 250, 300, and you look at the WHO estimate of 2,500, and you look at Chennai registered deaths of 3,000 per million, you know which number is likely to be correct. So lessons, we need to pay better emphasis on genomic surveillance in the future. We need much deeper integration with clinical data. We need integration of better diagnostics and everything else for actionability. In WHO, we have a warning system for emerging SARS-CoV-2 variants, but no system works if the data is not right. And no system works if the data is right, but you don't trust the data. This is an example what I would call good actionable surveillance. This is South Africa. And what you see is everything is Delta. The red is basically Delta. You see a little bit of something else, but pretty much everything is Delta until September, October 2021. And suddenly, in one province, on November 8, samples start coming in a different color, B.1.1.529, later on to be called Omicron. Just one province, one signal, caught in the same month, and trusting your own data. Then they have a cohort. This cohort undergoes regular testing, and they find that people previously found to be infected are getting reinfected at 2.5 times the rate of previous waves. So now they have two pieces of data, same month, 3 weeks. So I chair the WHO meeting, as was mentioned. Our meeting is around the third week of November. Samples are from November 8. We already have this much data. A new variant has completely displaced Delta in one province. we are seeing 2.5 times the expected rate of reinfection with this new variant. We decide to call it a new VOC, which is called Omicron, and the rest every one of you knows. And Omicron, of course, was the perfect immune variant. Every little lollipop on this graph is a mutation. Look at the one on the extreme right, Omicron. It looks like a brush. I mean, that's how mutated Omicron is. And of course, we did not see as many deaths with every subsequent wave we have had. You know, Omicron initially was BA.1, India was more BA.2. Later on, there was BA.4-5, and I'll come back to why we don't call them pi by now. Basically, what you see is that outbreaks keep occurring, but as more and more people have been previously infected and recovered, the number of deaths go down. Also, Omicron changes characteristic. Rather than going deep into the lung, it tends to go a bit more to the upper airway. And upper airway is less likely to kill you because of pneumonia, but might give you severe headaches, of course. And we must never underestimate the long-term consequences of these infections. Already we are seeing the cardiovascular risks, the thrombotic risks are significant. So immediate overloading of healthcare infrastructure problems seem to be diminished, but never take this as a innocuous virus. So what we do these days is something called antigenic cartography. And I'll explain this a little bit more on the next slide. We have a collection of sera in labs across the country, across the world actually. They know who has been Delta infected, Beta infected, Omicron infected, both infected, and you can look at neutralization. So if you are far away from each other on 2 points, it means neutralization went down a lot. If you're close together, it means neutralization, cross-neutralization is preserved. So what you see is gamma, beta, alpha, pretty much work together. If you were infected with one, you were reasonably good at cross-neutralizing the other. You can see Delta was a bit further away. You can see Omicron was a lot further away. But within Omicron, you can see that BA.1 and BA.2 were closer together than, say, Delta to Omicron. And the other important thing over here is that cross-neutralization is only one part of the thing. It's about infectability. Your cellular immunity protects against severe disease. So being further away here means that your risk of infection with that strain will rise. It does not mean that you will have severe disease. So keep these two things separate in your mind. This is a much better one where every 1 unit movement on the grid is equal to a 2-fold loss of neutralization. The graph on the right is perhaps more important because it has to do with vaccines. What you can see is vaccines remained reasonably protective, although you can see Delta is a little bit away from the cluster until Omicron came. And Omicron was really far away. On this particular graph, BA.2 is closer to BA.1. And you can see from a vaccine point of view, BA.1, BA.2 were not actually very different. They're right on top of each other. And this is more recent data on BA.2.7.5 that we were talking about. And this one is going to be a little difficult to walk you through. I want you to focus on the top middle graph of BA.1 breakthrough. It shows in boosted people, even if your original infection was BA.1, almost all of the 13 people have neutralizing antibodies. The effectiveness is down, which means given time, some of them will go below the neutralizing level. The neutralizing level is the dotted horizontal line. So with time, if you had a BA.1, you will break through that line and lose neutralization. If you had a BA.2 breakthrough, it will take you even longer to cross that line. But as of now, all current Omicron lineages, including BA.2.75, show at least modest cross-neutralization. And this is natural infection. Vaccines can give you higher titers than natural infection, which means our expectations based on antigenic cartography is that any vaccine built with any of the Omicron variants, including BA.1, which actually, as I showed you in the previous slide, is the furthest away, will give you broad enough neutralization response to block the others, which is the main reason we have not been in a hurry to create the next Greek letter pi, because it will simply create confusion in the minds of people. Are we getting an Omicron vaccine? Are we getting a pi vaccine? If all of them are cross-neutralizing, why confuse people when the purpose of the letter is to simplify things? We have complicated names that my colleagues over there and Dr. Karakarte use fluently to describe finer things. The best cartography is probably done with hamster sera, because every animal is born uninfected, you know that. You can keep infecting with every possible combination that you want, and again you can build these maps. And again you see that BA.1 is the furthest away on the antigenic cartography, and the broad response that you typically get, the typical size of the band is shown in the circles, will be sufficient to neutralize BA.2. BA.4.5 actually come back a little towards the center on this map. I have not shown that here. So again, there is no particular reason to worry. I'm going to end by telling you, it is going to be perhaps even possible to have a pan-sarbecovirus vaccine, a vaccine that neutralizes all variants. This is incidentally finding, I would say, people previously infected with SARS-CoV-1 who received the Pfizer SARS-CoV-2 vaccine are now creating antibodies that can neutralize every variant and every other coronavirus. So the right combinations are possible. So what's the way forward for all of us? We need to get a greater breadth in the immune response against circulating and emerging variants. Of course, our vaccines already work well against protecting for severe disease, but we should make them better. And we need to modify our vaccine composition to get this broad response. Do we already have techniques to do so? Yes, we do. So there are self-assembling nanoparticles with expressing proteins. You can put 4, 5, 6 different antigens on them. Already bivalent ones are available on the mRNA side. Bivalent on the protein side will come very soon. Killed virus vaccines are not very good for this, but again, it's not impossible to do that. By and large, the vaccine technology is rapidly advancing, and I look forward to the time when cheap, not requiring extreme cold storage, high quality, multi-antigenic, pan-sarbecovirus vaccines will be available for us all. Till then, of course, I will settle for SARS-CoV-2, but that is really all I wanted to say. Thank you all very much.
Thank you, sir, for mentioning our Maharashtra work in your lecture. And I am very happy that you have taken such an important topic. Now, variant, sequencing, and then vaccination. And when you deliver your lecture, you have take that science along with you. You have given your lecture in a very point-wise, in a sequencing way. So you have done a sequencing. Dr. Karakar already find out the variants in the Maharashtra and Delhi. What we discussed, that if it would have been done very previously, that is the tragedy in India, that we were late in doing the sequencing. That is the importance of sequencing. And still, from your lecture, we come to know that many people are asking in the public to me that why this sequencing, and why it is required. So from your lecture, I got the answer, and I think that even everybody is clear now that unless and until you do there are variants are coming up, you do the sequencing, vaccine will not be developed, and that is the common man's problem. Ultimately, we are for the common man. So if we do a good sequencing, good vaccine will come. Thank you, sir. Thank you for your good lecture.
Now I request Dr.
Sheetal,
Ingole
ek
sir
minute,
to
Sheetal,
felicitate
Sheetal,
Dr.
Sheetal,
Anurag
Sheetal.
Agarwal.
Sheetal, ek minute. It's a very important topic. And so we'll just have some questions, if at all, if anybody wants to ask. Yeah, it's a very important topic, and you are not going to get such a luminary ever together everywhere. And you have a chance to ask a question. So I request if somebody wishes to, yeah, you can. So somebody, yeah, come here.
Sir, I am Dr. Agar Khedkar. I am not microbiologist. I am a pediatrician. I am a MMC observer for this conference. I was part of vaccine development, and I did some trials. When we were doing studies in children, I found that 80% of children were already having antibodies.
Yes.
And we wanted children with swab negative and antibody negative children to be enrolled in the study. So during this Delta as well as first and second wave, many children were asymptomatic.
Yes.
They got infected. But they came up after 6 weeks with MISI, multi-inflammatory syndrome in children. and they had symptoms ranging from CNS manifestations to coronary dilatations. All sorts of complications were seen. So I just want to know what is the immunological phenomena in children which causes such havoc in their all systems, sir.
So a better person than me is available in this room who investigated the genomic basis of perhaps susceptibility to MIS-C, so Dr. Vinod Skaria. So perhaps during his talk he will touch upon it. But it seemed to be a sporadic event. Genetics would explain only a small fraction of it if I understood Vinod's data correctly. And we are not very clear why it happens, except pretty much if you look at any viral disease and you look at rare manifestations of MIS-C-like syndromes in children, they've always been there. What we saw was a short period in which millions and millions of children got infected. So what normally appears to be rare suddenly became commonly seen. And I think the same is true for deaths. If you actually look at the death rate from SARS-CoV-2 infection, it is not more than 1.5 to 2 per thousand. It is not. It is simply that we see the tip of the iceberg in terms of the infections, and we look primarily at, so in our institute, when we follow by antibodies our entire cohorts across India, 60% of the infections, even in adults, because we have no children working in our institutes, are asymptomatic. With children, it is almost 90%. So yes, it's just which part of the iceberg you see, depending on which age group you treat.
We can take, yeah, one more question we can take.
A very excellent and lucid talk. It was really very nice to know about these variants and all other types. What we wanted to know, in this very city we have NIV, and recently when we got the monkeypox cases, just because of this genome sequencing, and then one of the scientists told that we all are having all kind of viruses in India especially, be it the monkeypox, Zika, and so what will be the future of InsactCog? We have started with SARS, but do you think after 2 years down the line, we really needed to strengthen with other types of viruses also, to include the other viruses also?
No, you asked a fantastic question. So there are two ways to learn about something that is circulating. One is you go after all cases to find out what is happening, or you wait for something to become so overwhelming that you are forced to find out what is happening. More often than not, we follow the second principle, that a sporadic death here and there is not taken seriously. In fact, I'll give you a comment that will help illustrate this. Somebody came back from his village and we were talking about, Delta wave, and he said, nothing unusual happened. Yes, this time old people, when they got sick, they died. It was fine otherwise. So it's a question of, are we investigating the cause of each death? Is there a culture to track down things that are unusual and look for causes? I can be certain that if we were to do that, we'll find plenty of viruses apart from those first being described outside India, then being tested for inside India. We'll find many more things in this world that are being found for the first time inside India. It is not that we have a shortage of unexplained deaths when you start looking at especially those parts of our country that are very rural, in connection with forests and so on. And I think it will take public health, microbiology, possibly a One Health approach to all come together to answer this thing. And the more we find, the more we look, the more we will find. The more we will find, the more we will learn. The more we will learn, the more we will be able to prevent these things from occurring in the future. But we are at the very, very early stage of this knowledge progression, and the work being done here of course, is a fantastic step forward. Thank you.
So, the last one? Last one?
Of course, of course.
This is the last one, huh?
I'm just
Please.
trying to get them to lunch.
After the vaccines were discovered in 2021, so there was a trend of measuring antibody titers in general population who were not even infected again. Because people came to know about that after the vaccines, if you want to measure how effective is the vaccine, So we have a test known as an antibody titer, in which you can see how effective the vaccine is. So there were people, relatives and other people coming to us and asking that they have taken Pfizer, they have taken Covaxin, they have taken Covishield. But the range of antibody titers is different in different people. And they were not up to the mark because we were having more than one and less than one as the report for. So they asked me the question that how effective are the vaccines, because up till now we have not been infected, but my antibody titer is not high enough to protect me from the infection. As compared to the blood viruses, for example, hepatitis B, which we have taken in our childhood, but our titers are still very high. So this is something related to the variants of respiratory viruses, because the respiratory vaccines are not effective, or it is something else. That is what I wanted to ask.
It's a great question. It's going to take a little bit of a longer answer, so please bear with me. When you plot efficacy of a vaccine versus the antibody titer, it actually follows a very nice curve. You can back extrapolate the efficacy. When it comes to a new variant, what happens is a loss of neutralization. So if you started with very high levels, you are towards a plateau and you fall back, you are still at a high level. If you're already on the steep part of the slope and you fall back, you fall to below neutralization. This was very obvious with Delta. So for example, if effectiveness against index variant, we don't call it Wuhan anymore, effectiveness against index variant was in the 80% range for all the vaccines. It was in the 90% range for mRNA. Then came Delta. And we know that antibody production was the lowest for the killed virus vaccines, higher for the adenovirus, highest for the mRNA vaccines. When Delta came, the mRNA went from the 90s to the 80s. In our own data, Covishield went from 70, 80 to about high 50, 60, and Covaxin went to about 55, against prevention for infection. Prevention of severe disease was reasonably good for all. Then came Omicron. The mRNAs went down from 70, 80 when they reached during Delta, down to the 50s. Others went to pretty much not working at all. When the same vaccine was given in combination with previous infection, now the efficacy was much higher because the response was broader and the response was stronger. And we must keep in mind that some of the data that claimed only a 3-fold reduction of whole virus vaccines with Omicron, the patients were selected in October, November 2021 from the National Capital Region, and the decision for infection or not infection was made based on history and presence of RT-PCR tests. I showed you that India, Delhi, NCR was 90% infected by then. So you would imagine that in that particular study, if they did not actually check for antibodies to rule out infection at the beginning of the study, they must have given vaccines to previously infected people. And that only 3-fold loss of neutralization is probably an artifact stemming from that the reviewers of foreign journals don't always know regarding what happened in India. Now coming to your last implicit question, do titers matter? Yes, they do. Pretty much line them all up, that's the way they have worked. I'll give you one last example before I stop in Hong Kong. BA.2, which is Omicron, was felt to be very innocuous in India, right? But India was already 80-90% previously infected, almost everybody was vaccinated. Hong Kong was 10% previous seropositivity at the time Omicron came in, and only 1/3 of people above 80 were vaccinated. Of those 1/3, most had taken the Chinese killed virus vaccines, not the mRNA vaccines, because physicians told them go with tried and trusted safe technology, don't go with this newest technology. To me, that's a particularly poorly thought through statement. The oldest person has the least to worry about long-term complications of an untested technology. It is the youngest person who actually is the most to worry about. I would have done the other way around. Give young people killed virus vaccines, give the oldest people mRNA vaccines. When you look at the death data in Hong Kong, which was presented to us, you can see pretty much everybody who died, not everybody, but almost everybody who died, were the ones with the killed virus vaccines. So fundamentally, we have to have all vaccines are good in general. All vaccines have good applications. If I had to choose a vaccine for my daughter in the 2 vaccines available in India, I wouldn't even have thought for a minute before saying Covaxin is the correct vaccine for my daughter. As a young person, low risk, and with thromboembolic phenomena or high. But we need to remember that things are different. We must interpret all things carefully and not get it caught into wide ideas that everything is identical. Hopefully I answered your question.
Thank you, sir. You can have your seat. Yes.
Now I request Dr. Ingole sir to felicitate Dr. Anurag Agarwal sir. I request Dr. Reshma to felicitate Dr. Ingole. Now we will break for the lunch. I request you all to take your lunch coupons for the lunch. The lunch arrangements are done in Pandal and other arrangement is next to Dean office. Thank you. Our souvenirs are at the registration desk. Please collect your souvenirs. Also, our poster presentation will be starting soon. I request all the presenters to display their posters on the first floor. Thank you.
Now I request our guest speaker, Dr. Sridhar Sivasibbu, to come on the dais. I request Dr. Rashmita to introduce Dr. Sridhar.
A very good afternoon to all. Dr. Sridhar Sivasubbu sir is a senior principal scientist and head business development at the CSIR Institute of Genomics and Integrated Biology, Delhi. He is also a professor, Faculty of Biological Sciences at Academy of Scientific and Innovative Research. He completed his PhD from MS University, Tirunelveli, India, and did his postdoctoral research from CSIR Center for Cellular and Molecular Biology and the University of Minnesota, USA, with specializations in genetics and genomics. His research interests are focused on sequencing human genomes for adoption and benefit in healthcare with applications in rare genetic disorders. He also develops zebrafish models for application in precision medicine in humans. His laboratory also explores non-coding RNA-mediated regulation of vascular development and diseases, wherein his group is credited with the discovery of a number of non-coding RNAs. His group also piloted and leads the genome sequencing-based surveillance of SARS-CoV-2 for India and Southeast Asia. Sir has co-founded the Genomics for Understanding Rare Disease, India Alliance Network, that is Guardian, which is one of the largest precision medicine ecosystems in India working on rare genetic diseases. He also served as the Chief Scientific Officer of the Center for Genomic Application, a public-private partnership company established by IGIB. With an elaborative academic career, Sir has 141 publications within his specialty, which are published in reputed national and international peer-reviewed journals. It is an honor and our privilege to have you with us today. We welcome you, sir.
Good afternoon, everybody. I know it's post-lunch and the lunch was good. So I know you'll all be feeling a little bit relaxed, but I'll try to make sure that you enjoy the talk also. So a good thing about this, my talk is that between Dr. Rajesh Karyakarte and Dr. Anurag Agarwal, I think they have pretty much covered most of the highlights. So what I'm going to do most is trying to tell you what happened in the background. Okay, how did we go about doing this? So what you already know. I'll talk about the how, and I'll also try to lay down its significance going ahead. But before I do that, you all since morning, Dr. Karyakarte really talked about IGIB, and I wanted to let you know that's IGIB. And if you are ever in Delhi, we are in the southern part of Delhi. Please feel free to visit us. We would be happy to host you and give you a tour. We are a very small place, about 42 scientists, and core strengths are in disease biology and translational research. We take extreme pride in making sure that our faculties are interdisciplinary, and as you will see, that's one of our key strengths. So with that introduction to IGIB, let me move on. We have heard about this paradigm shift in microbial characterization, which has come, it was there always. It's not that as if it has only come in because of COVID, but COVID did amplify this to a large extent, wherein instead of doing the traditional microbiology way of looking at an organism, you could just subject it to a large-scale genome sequencing and be able to use informatics in a smart way to extract and figure out what is the pathogen or the organism. And largely this is because of the technology as well as the cost reduction in the cost. As in the case of COVID SARS-CoV-2, you could take a nasopharyngeal swab, extract the RNA. We saw that number of talks today. Use informatics to find out what is causing it. But one of the key tricks to why we could use informatics so excellently in SARS-CoV-2 is because of this genetic variability that it periodically introduces, which Dr. Karyakarte actually introduced to us with the various nomenclatures. The nomenclatures essentially are a reflection of the genetic variability that this virus accumulates in a very periodic manner. So you are able to do this nice detective work of going back and forth and figuring out what is the variant that's out there. So what does genome sequencing do? It helps you in understanding the evolution, which we heard today. It helps you in confirmation of the virus, and it helps you in identifying origins of the virus, but also it helps you identifying, evaluating the diagnostics, vaccine development. We have seen all of these in the morning first half, and I'm thankful to my speakers, but all this could happen largely because of one key stepping stone in genomic research. Okay. And so we in CSIR around early March 2020, when India was still under the first lockdown, sort of sat down. We have all the ingredients here, Dr. Anurag Agarwal, Vinod Skaria, myself, our head at that time, Dr. Shekhar Mande, who also incidentally happens to be from this city, or was in this city for quite some time as the head of NCCCS. Okay, sort of realized that the single RT-PCRs are good. They do positive and negative detection. But going forward, the way this virus is mutating, you will need large-scale sequencing exercise, and we call this the mega lab. Okay, and this has to be there in almost all cities in India, so that you could keep the businesses open. We could track the virus, and this was, remember, in March 2020. when people are trying to grapple with what does this virus even do. So I think this is a visionary leadership of CSIR that really paved the way to getting the first global field trial of this assay called as COVID-seq. Illumina had just launched it 2 weeks ago, and we were one of the first recipients of this globally. And what Dr. Vinod, myself, and our team that's down there at the screen did actually was to pilot this assay, and the way the assay was given to us and what went out of our lab are completely two different protocols. But nevertheless, this protocol got adopted over and over again, to the extent that today the default technology internationally is COVID-seq for genomic surveillance of SARS-CoV-2. And you can see the outline of the technology is pretty complex, but I'll try to break it down that it's nothing more than getting the RNA, subjecting it to genome sequencing, and doing a lot of smart informatics around it. So with that, the first field trial that we did was from 7 states in India for about 2,000 genomes, 3,000 isolates, and out of which came out 2,000 genomes. There were duplicates in the samples just to ensure that reproducibility and everything was maintained. And that became internationally the first data set for COVID-seq before it was subjected to a lot of scrutiny. So what it told us was you could use sequencing for detecting the virus. You could have a scalable configuration. You could go from 750 to 3,000 samples, and I'll show you later on how we could go from all the way from 24 to 3,000 samples. That's a little bit of protocol development that we contributed. It in many ways had better sensitivity because it could look at single nucleotide resolution. It could give about 10% more diagnostic yield, but most importantly, it could reclassify about 50% of the samples that were pan-Serboco labeled into actual COVID positive samples. So early on in the pandemic, if you remember, people who are pan-Serboco positive, there was a doubtful case. Did they really carry the virus? Did they not carry the virus? So we really showed that they do carry the virus. So but that gave India the opportunity to become global leader in the genomic surveillance. So today all the success that you see around INSACOG, what has happened in Maharashtra, in the other states, is largely because of this one single visionary leadership that we had in Dr. Shekhar Mande, Dr. Anurag, and my colleagues at IGIB that allowed us to do this fantastically. So just to quickly go through what you already know in many ways. Early on in the pandemic, as you will see, that the bottom screen, at the bottommost of the screen, there's a circle which shows the 2 introductions from the original viral strain from China that came to Kerala. But then you saw this nice blue color, that large blue color that you see. Each of them is a single viral sample. You could see that they were completely different. They had this mutation called D614G, which became a defining mutation in them. And if you looked at the spread of this particular variant, which is called as A3I or A2A, depending upon the time scales that you looked at, it was spread large across in India in green color, in the purple color, and the green was the A2A. And this really gave us the first landscape of the distribution of this virus in India. But what we quickly did, which Dr. Karyakarte mentioned about, was we partnered with the government of Kerala to actually start asking interesting question. Is the virus that is circulating in Kerala is partly because of international introductions into Kerala, because Kerala happens to have one of the largest expatriate populations coming into country, especially when the lockdowns happened. So in such a scenario, the question was, is it because of the international travelers, which one would expect? But it was interesting to note that it did not happen due to international travelers largely, because there was a very good quarantine system being maintained. It happened largely due to local outbreaks, and the outbreaks happened largely because of the land borders with the neighboring states. So this was one of the first instances where public policy decisions were being taken based on genomic evidences. Okay, so this gave us inputs that if we continue doing this genomic surveillance, we should be able to contribute to public policy decision making within the government. So based on this, the land borders were tightened up at many places, and they were able to figure out how effective it is on a week-to-week basis looking at the genome sequencing. And that led to this program called as GeneScope2 with the government of Kerala, which is a very successful program, heavily used and cited internationally. And some of the public policy decisions are on this slide here, right from early tracings introduced by Kerala government to the double masking to statewide lockdowns. All these were decisions which the bureaucracy or the political establishment could take based on scientific evidence. That was encouraging to note that. So we could also put out maps like this which helped them make decisions. These are called phylogenetic maps. But following closely up on the Kerala model, Dr. Karyakarate alluded to this, of the genomic surveillance that was partnered between the state of Maharashtra and CSR IGIB. And here I would like to thank both Dr. Shashank Joshi and Dr. Shekhar Mande, who were visionary enough to think through this process and felt the need for it. So we started this program, and this is the first data that you see out of Maharashtra for June 2021. And every district, and I should really take this opportunity to thank every district coordinator. Over a 1 year period, I think on a weekly basis, everybody religiously sent us 100 samples. That is 25 samples every week, every month 100 samples got shipped out, delivered. And that kind of discipline, I should really appreciate and give all of you a big hand. Can you have a round of applause for all of you? I think that kind of discipline rarely seen, and Dr. Rajesh Karekarte, thank you for leading that fantastic collaboration. But having said that, this actually Dr. Karekarte actually presented, so I will not spend too much time, but we could capture the evolving landscape of this virus over a 1 year period, April, May, and you would see the colors changing, nothing but the virus changing. And it was really good to see that Maharashtra bureaucracy and the government using that to take policy decisions. I remember both the incidences that happened in Aurangabad, the Ratnagiri and the Sindhudurg incidences where we actually pioneered this accelerated protocol. And this excellent postdoc in my lab called Rahul Boyer, who also happens to be from Maharashtra, actually spearheaded this particular protocol, which really led to the AY1 and AY2 variants from Ratnagiri being identified. And of course, we heard about the Delta and Delta lineages in extensive, so I will not talk more about it. Rather, I'm going to skip a few slides and sort of summarize this. So what happened in the state of Maharashtra was a global picture of the state from a distant, trying to use that information for public health benefit. And again, this is something new for a scientist like me. The reason being, we do a lot of discoveries, we publish papers, we talk to clinicians, but we have never talked to politicians and bureaucrats. That's something that is one step away. Okay, we write notes and give them summaries. What happens to those notes, nobody really knows. Okay, but here on a weekly basis, if the chief minister is sitting on the table or the chief secretary is sitting on the table asking questions, doubts, as if they are the student and we are the professors, we had a very refreshing experience. And I think that experience, I wish it continues in the Indian public systems, because there are a lot of problems that require such solutions. But anyway, the CSR IGAV being one of the founding members of our INSACOG, with the leadership of Dr. Anurag and several others in the country, have put this infrastructure in place. I hope that they continue this model into other diseases also. But this is one slide that I would like to leave you with. Very important, probably the most important slide in my talk. Does genomic surveillance really help, or is it catching up game that we play just to please the record books? So if you look at this, okay, there is a the peak that you see on the top right hand side of the graph, the top graph, is the wave that we really see in Delta. The bright red dot that you see is the outbreaks that happened, first outbreaks that Dr. Rajesh Karyakarate reported from Yavatmal and Amravati. Okay? But can you now play a detective game and figure out when did Delta actually begin in this country? Okay? There is a green triangle there in August 2020. That's where the first signatures of Delta showed up. Okay? So now for genomic surveillance can tell you almost 6 to 8 months in advance that you are going to have a variant. And if you notice Dr. Anurag's talk, where now we have science at a pace where you can predict a mutation, and they could almost tell you what this mutation, will it be a severe one, will it neutralize, will the vaccines work? Okay. Now combine these two together, you have a very potent surveillance mechanism and prevention mechanism. So that 8-month window should be used in a very, very effective way. That's the message that I would like to say. As microbiologists, you should focus on figuring out, not ignoring that one outlier that shows up on your data sheet. Okay? That outlier is probably what's going to lead to the next big outbreak of whatever pathogen you are looking at. So my request is, please keep an eye out for that one outlier in your data sheet and flag it up. Okay? And Vinod will probably tell you about, end his talk probably on how you could communicate with us if you have such outliers. At IGIB, one of the key things that we propose to continue is to not leave this momentum, rather look at these outliers very closely and keep a tab on them, not only for SARS-CoV-2, but for any emerging infectious disease that we occurs in India. As a public institution, I think it's our responsibility to respond. If you have the knowledge and the resource, we should do it, and your corporation is very much welcome on it. Okay. We did talk about this accelerated sequencing. Now, I'll give you one or two slide example of how this was used. So this was a technology that we came out of necessity because urgency was important. You had a plane load of people sitting in the Delhi airport wanting their data. You could not wait for 1 week. Okay, or you had a hospital full of people, a ward full of people, or a mohalla full of people. You wanted answers. So we had this RNA to sequence that could go in 24 hours. And here are some of the examples. So Dr. Balram Bhargava, who was the head of ICMR, actually wrote to us because there are 38 deaths at Aligarh Muslim University in about a week's time. These are all faculties. So we wanted to figure out what was circulating. Same happened with the Ratnagiri and the Sindhudurg areas where there was a sudden new variant seen. Or for that matter, now what we have done is we have partnered with WHO, and this is the default assay for WHO across 11 countries now. So we get samples on a regular basis, we sequence them, we update. For example, here with Nepal government, we were able to really help them out on figuring out what is the virus. We do that for now all the CRO countries, that is 11 countries that WHO handles. The key learnings are that genomics does provide enhanced sensitivity and diagnostic yield. It does allow you to have a very efficient surveillance system. You can do both high throughput, and you heard both the Illumina as well as the nanopore data today, that these technologies are very powerful. You should use them. But most importantly is that collaboration between scientists, policy makers, political establishments to make public health decisions. That connect was very powerful. And I'll stop there for the genomic surveillance, but then move on to something very different that we did. When you have a lot of data, what do? Can you use that to better our diagnostic kits? Okay. So what we did is, because we use RT-PCRs and everything, sometimes RT-PCRs fail to work because there is a binding problem of the probe, because there is a mutation there. Okay. So between Vinod's lab and my lab, we kind of map all the kits that are out there, trying to figure out what is the best way to design a primer probe, and out came this very nice kit that we call as genome proofed RT-PCR now, which is manufactured by Siemens. And I would like to thank the two students who came up with this idea to do this, Gyan and Paras in my lab. But we use these things for not only for surveillance, but you could also use them for looking at persistent infection, reinfection, reactivation, or breakthrough infection. All these are common things that will happen in any large outbreaks. So here are examples of one of the first, I think this was the third case of reinfection internationally that we documented. This was out of the GIMS Greater Noida with Dr. Vivek Gupta as our collaborator, where we were able to document the reinfection case. We were able to look at a reactivation case in a child with neuroblastoma, again from a hospital in Delhi. So you can do a lot of tricks other than just looking at surveillance. So, and this is important because as microbiologists, these are things that would keep you interested in solving these puzzles. We were also able to look at breakthrough infections in healthcare workers in Kerala in a large hospital and figure out how the virus breaks through the infection, characterize the virus. So largely you could do a bunch of nice informatics to derive these answers, and that's a very worthy of the time that you spend. But as IGIB, like I said, our goal is to translate knowledge into products. So we also worked with a lot of companies to develop RT-PCR kits. A couple of my colleagues, Shobik and Debo, many of you have heard about this paper strip-based diagnosis called Feluda. I'm very proud to have been their colleague in this assay. Dr. Rajesh Pandey and my team at IGIB really spearheaded this genome sequencing using nanopores called the microlabs, okay, where you could deliver results instantly at the airports. And you can see the team of Lipi, Bhupesh, Rakesh and Rajesh Pandey who really spearheaded that. So a wide range of activities, not only about genome sequencing, everything around genome sequencing is what we did. And I'm going to stop there and acknowledge a large number of people here, probably Rahul and Paras, who led the teams. Vinod, who is here in the audience, who was a fantastic support and collaborator all throughout 2 years, and two people of key interests, or without him, the program would not have happened. Is Dr. Anurag Agarwal, you heard him talk today, and Dr. Rajesh Karyakarte. Thank you for your leadership, and of course, Maharashtra government, DMER, and many other state governments. I'll stop there. And lastly, I would like to thank the leadership of CSIR for their visionary guidance, as well as believing in us. Remember, I am a human geneticist, okay, not an infectious disease biology. So when we start talking infectious disease, it is like you have read something in the newspaper, so that's what you're talking about. So having believed in being able to deliver a technology is something that I really like to thank everybody in this room as well as my own leadership. Thank you. I'll take questions. Yes, sir. Please, sir. Can you get the mic working?
Hello. It was a very elaborate and very detailed account of surveillance and in reference to genomics. I would like to ask myself a question to you. India is vastly represented by rural masses, and bulk of the human population in India is based in rural setting. What is there any feedback available to you whether because most of the COVID related work has happened in cities and semi-urban settings, where the laboratories are there, RT-PCR is there, and even genomic sequencing facilities are there. Bulk of the populations who are staying in rural areas, we do not have exactly detailed availability from either public health department from various states. And do we have any such access to such information available from rural representative of the Indian population? And what is the status of the COVID virus spread among the rural population? And does the genomic sequencing and analysis provide us some clue regarding the transmissibility from the rural to suburban, from suburban to urban? Is there any track that we can trace, or whether the rural populations have remained little aloof from this happening? I would like to know from you.
Okay. Good question. So the question is, was there a difference in the genomic signatures that we noticed in urban cities versus rural areas? And that's the summary of the question. So, while I don't have very strong data, but given the fact that the mass scale migrations happened between the cities and the rural area early on in the pandemic.
Migrant workers.
Yes,
were
migrations
the
happened.
population. Yeah.
The virus has sort of also been pervasive both in the, initially it happened in the urban areas, but eventually it did reach the rural areas, maybe not at the same time scale, but in a lag phase, with a slight lag phase. So I don't necessarily think that they have remained aloof. Then at this point, I think everybody is equally infected, and but there might be a time gap, slight time gap. That's all. Thank you.
Thank you.
Thank you.
Any other query anybody has, he would be glad to answer. Dr. Ingole would like to. Thank you. Thank you so much.
Thank you. Thank you, everybody.
Thank you, sir. Now I request Dr. Jahangir sir and Dr. Ingole sir to felicitate Dr. Sridhar.
May I request you also on the stage?
Rajesh, yes.
Come.
Here by you.
Now I request Dr. Mayuri to felicitate Dr. Jahangir sir. I request Dr. Srishti to felicitate Dr. Ingole sir. Now I request Dr. Vaishali madam to continue the next session.